Hey everyone, we are going to be looking at reproductive growth and development today. Now, some of this we have covered in previous lectures, so it'll be a little bit of a review, but we're also going to go a little more in depth into certain topics. So, before we get to it, let's review. Okay, so last lecture we looked at germination, vegetative growth, and phase change. And we started with germination, and this is something we've talked about throughout the course. And we said that for germination to occur, we first need to relieve that dormancy condition that we've talked about. And then there are three important factors: temperature, the right temperature, water, and oxygen. And if you have those three, and if your dormancy is relieved, your plants will germinate. Now, for certain plants, sometimes there are additional requirements. And we looked at plants that required fire, such as those pygmy pines or smoke. But generally, the temperature, water, and oxygen are the big three. And then we also again brought up the different patterns of germination: epigeel, above ground germination, typical of our monocots, and hypogeel, below ground, typical of our dicots. And then from there, we moved on to vegetative growth, and we looked at the three main types of growth patterns that we see. We have our annuals, which go through their entire life cycle in one growing season; biennials, that go through their life cycle in two growing seasons. A lot of times those biennials require vernalization, that period of cold, in order for them to flower. And then lastly, we looked at our perennials, which live more than two growing seasons. And within our perennials, we had our herbaceous perennials, where typically the shoot system will die off in the winter, but the root system will remain alive and produce an entirely new system the next growing season. And our woody perennials, which maybe the entire system remains alive, maybe we'll just see a loss of leaves in the winter, etc. And we ended by talking about phase change, which is essentially plant puberty. And that is the shift from the plant from it being a juvenile to a mature plant. And what marks a mature plant is its ability to reproduce. When we talk about our angiosperms, that's the ability to produce flowers. Now, when phase change occurs, we also may see a corresponding morphological change, right? Some of our plants, you see a definitive difference in their morphology and their growth when they are juveniles as opposed to when they are adults. A good example is English ivy: different leaves, different growth pattern. And then phase change can be important from a propagation standpoint, because certain plants, if we are propagating by cuttings, can only be propagated when they are juveniles as opposed to mature adults. Okay, so in today's lecture, we are going to be covering these 7 topics, right? We're going to start with flower induction and initiation, what causes our flowers to develop; flower differentiation and development; pollination; fertilization; fruit setting; fruit growth and development; and then we're going to end with fruit senescence. So again, these are some of these topics we've already looked at, right? We looked at pollination, fertilization. We've looked at fruit development. So some of that is going to be review, and we won't go too in depth. But in other cases, such as when we talk about flower induction here in the beginning, we're going to delve into it a little bit more. So when a plant is mature, and remember from last lecture that maturity means that it went through phase change, puberty, and now it's reproductively capable. Right? So when a plant is mature, a change can occur in its apical meristems that switch them from being vegetative to being reproductive. And when we say reproductive, that's going to be forming flowers. Now, most of our agricultural plants are self-inducive. Right, so they are going to form flowers after they reach a certain morphological maturity. Right, some level of maturity they're going to reach, and it's going to trigger them to form flowers. Now, people often use a calendar to predict plant development for specific management decisions. Right, you've all probably seen on like backs of seed packets, whatever the plant it is that you're growing, it's going to take this many days to maturity, this many days to fruit set, whatever. However, research has shown that measuring the heat accumulated by the plant over time provides a more accurate physiological estimate than just counting calendar days. And the reason for this is what we've learned before, is that temperature regulates metabolic processes, which means essentially temperature is going to affect how fast our plants grow and develop. And remember, the warmer it is, generally our plants are going to grow and develop faster. So we can measure what are called growing degree days. And what this is really just a way of assigning a heat value to each day, and then we add those values up together. And in doing so, we're going to be able to get an estimate of the amount of seasonal growth your plants have achieved, as well as predict the development stage that they are going to be in, including flowering. So there are two methods of doing this. There's the temperature averaging method, which we're going to do, and then there's a little more advanced, complicated method called the Baskerville-Ehman method, which is technically more accurate, but it's a little bit harder the method is harder. I believe it, nowadays you can just use the computer for everything, but the actual to calculate the method takes a little bit more advanced math. We're not going to get into it. We're going to go with the much easier temperature averaging method. So how does it work? It's very, very simple. For each given day, you take the maximum temperature that was recorded that day, add it to the minimum temperature that was recorded that day, and divide by 2 to give you an average temperature. And then you subtract from that a base temperature. And the base temperature is the temperature below which development stops for that given plant. Now, the base temperature will vary based on the specific plant. Remember, right, each of our plants has this range of tolerance for temperature which it will grow within. So there is a little bit of variation amongst our plants as to the low end of that range, right? So below which they won't grow or develop. So you'll have to look up the base temperature for your plants if you're doing this method. So, for example, this is how it would work, right? The base temperature for corn is 50 degrees Celsius. So below 50 degrees Celsius, no development is going to occur. No growth is going to occur with your corn. So if we have a hypothetical day and there's a high of 83 and a low of 61, and actually, I'm sorry, this is incorrect. This should be 50 degrees Fahrenheit. 50 degrees Celsius is super hot, I'm pretty sure. So 50 degrees Fahrenheit. So you have a high of 83 and a low of 61. How many growing degree days did you accumulate for corn on this day? Well, we take the maximum temperature, 83, add it to the minimum, 61, that equals 144. We divide it by 2 to get an average for that day, which was 72 degrees Fahrenheit. And then we subtract our base temperature for corn, which is 50 degrees Fahrenheit. And that gives us 22. So for that day, our corn plants accumulated 22 GDDs, or growing degree days. Now, how this is helpful is when you realize, or when you know, that it takes around 1,000 growing degree days for flower induction in our corn. So if we're tracking this each day, it's going to give us a better estimate of when our corn, is going to go through different developmental stages, right? So it could be helpful for that management, planning things out. So, like I said, there's two methods for this, and the temperature averaging method is the easier method. But why we may want to rely on the other one is that there are some issues with this temperature averaging method. And one of the big issues is that it will underestimate growing degree days when your temperatures are low. So how does this work out? Well, let's give an example, right? So for here we have an early May day, and our max temperature is 57 degrees Fahrenheit, while our low was 42. So if we use the temperature averaging method, we would take our max temperature, 57, add it to 42, gives us 99. We divide by 2, which gives us 49.5, and then we subtract our base temperature of 50, and we're left with a negative 0.5, or if there's negative here, we just calculate that as 0. So if we were using this temperature averaging method, for this day, we would say that it accumulated no growing degree days, right? However, at least for a certain portion of the day, the temperature was above 57, was above the base temperature of 50, right? So for at least a little bit, this plant was likely growing. Because of that, it's when your temperatures are low, so earlier in the season or later in the season, you're going to underestimate or not include these small cases where, at least for part of the day, your temperature was above your base temperature. How much of an effect that is on the accuracy, I think, is something that could be investigated. I'm not sure how much it would really affect things. However, if you're really a stickler for accuracy, and maybe depending on where you're located in the country, this may be more of an issue or not. Okay, other things to notice with our growing degree days is that, as I talked about in a previous lecture, generally your crops don't grow any faster at temperatures above 86 degrees Fahrenheit. Right, so that is your max temperature, even if it was hotter in a day, right? So even if your max temperature for a day was 92, you're still going to use 86 as your max in the calculations. Now, like I said, growing degree days is really neat because with them you can calculate or at least estimate when your plants are going to reach different stages. And it's not just flower induction, but it ranges the gamut from emergence to stem elongation when you're going to detect the first nodes, when it's mature, when the seeds begin to fill for different species. Right, so it's helpful from that management standpoint. And you can also calculate this in both if you're using Celsius or Fahrenheit. Now, besides plants, growing degrees are helpful calculating them because insect growth and development is also predicted by growing degree days, right? You can also look up similar charts that you can see here. You can look up for different insect and insect pests. And using this knowledge, and if you're keeping track of this, you can predict when certain pests are going to be a particular problem or when they're going to be most vulnerable to some sort of mitigation, whether it is using pesticides or some other kind of targeted pest control. Now, that is a majority of our agricultural plants are self-inducive, right? They are just going to produce flowers at a certain point. They're going to reach that reproductive maturity at a certain growing point. But other plants will flower or show changes in development in response to variations in day length. And we've looked at this before, right? When I talked about how light affects growth and development. And this is referred to as photoperiodism. And if you remember, we have three different types of plants. We have our day neutral plants, right? That would be those agricultural crops that aren't responding to variations in day length. They're self-inducive. And then we have short day plants, which will flower when day length is shorter than some critical day length. And long day plants that will flower when day length is greater than some critical day length. And if you recall, that critical day length is different for different species, right? It's not a standard measurement. Now, what's interesting is when they started investigating this, it's actually the length of night that makes most of the difference, right? So they originally thought it was day, and it's why it's named this way. But if you really look at it, if you take a short day plant and you are growing your plant, and here's the critical period right here. So it would be below the critical period in terms of light. But while it's growing in the darkness, you give it a flash of light at a certain point, it's not going to flower, right? So you see here, this is as the day lengths are decreasing. Here where you're past some critical day length, and if you go throughout the night, it will flower. But if you give it that flash of light, it's not going to flower. And the same thing, the opposite is true for your long day plants, right? You could have a length of daylight past this critical day length, and as a result, your long day plant is going to flower. If you don't have it past, it won't flower. If you don't have it past, but then you give it a little bit of flash of light here during the night, it will flower. So what it seems is it's actually length of night that is more important. But because we originally referred to these and thought it was day length, that's how they're continued to be referred to. Some other interesting things about photoperiodism is that the leaves are critical for this to occur, right? So if you remove the leaves of a plant, they will fail to flower, even if you're giving that plant enough light to induce flowering. And vice versa, cocklebur, which is a short day plant, was exposed to long hours of light, but it still flowered if you blocked light from a single leaf. And what this suggested is that there is something happening in the leaves that is then translocating to the apical meristems to then cause flowering to occur from the apical meristems, right? They have that switch from vegetative to reproductive. But they didn't know what it was, and they thought it might be a hormone, and they called it florigen. And people searched for this hormone for 70 years. They could never find it. What was happening? Well, they have recently come up with or have been able to figure out at least the method while how some plants, excuse me. Well, recently they have worked out the mechanism of photoperiodism for at least some plants and for some long day plants. I am not going to require you to know it because it is very complicated. I am going to teach it to you, right? I'm going to give you a kind of a brief overview of what's happening. And it involves those transcription factors that I talked about before when I talked about genetics, right? Those proteins that turn genes on and off. It involves a whole ton of these transcription factors. So I'm not going to require you to know it, but I want you to see it just to get an appreciation of really the complexity of what is occurring in these plants. It's a very complex process that also interestingly arises from a relatively simple system, right? All organisms have this simple system where it is 4 nucleic acid bases that make up our genome, and it's the arrangement of those bases and then all this complexity that gets built into it from those simple bases that produces everything, all biological life that you see. So it's more for you guys just to kind of get an appreciation for this process. I'm not going to require you to know it, but let's take a look. Okay, so the mechanism of photoperiodism has been outlined in some long day plants. And I'm going to go through it here. Again, you don't need to know this, but I just want to provide it to you so you get an understanding of the process and kind of what I find interesting about the process because it's so complex. But at its core, what we find is that the induction of flowering is the result of a gene, flowering locus T gene. And what happens is the more that this gene is expressed, the faster that the plant is going to flower, or it accelerates flowering. So what this means when I talk about gene expression, that's a process where we go through transcription and translation, where we go from a segment of DNA to create a protein, right? So this gene here is found in the cells in our leaves, right? And it gets expressed, so it starts making protein. We'll just call it FT protein. And that FT protein travels to our apical meristem, and it results in changing that from a vegetative bud to a flowering bud, and that initiates flowering. Okay. Now, what controls this gene expression is CO protein. So there's another gene, a CO gene, and I'll say CO protein / gene controls FT expression. So this CO protein is a transcription factor. It turns this FT gene on in this case, right? So CO turns on FT gene. So this CO protein turns on this FT gene. That FT gene, once it's turned on, it starts making this FT protein, and that FT protein is going to result in flowers. Now what's happening is that this CO gene is affected by a number of other transcription factors, and those in turn are regulated by the circadian clock of the plant. So just like in humans, we have a biological clock that kind of regulates us. We have different processes that occur in the morning versus the night, right? Same thing is occurring in our plants. So CO gene is regulated by other proteins and transcription factors, which in turn are regulated by plants' circadian clock. And what keeps that plant's circadian clock are different light receptors. Just like our phytochrome pigment system or the phytotropin, there are light receptors, primarily blue light receptors, that maintain this circadian clock in our plants. And some of them are called cryptochromes. So to get more specific, what's happening is in the morning, there are other transcription factors that are produced called cycling DOF factors, or just CDFs. And these CDFs prevent transcription of CO gene. Right, so these CDFs that are produced in the morning prevent the transcription of the CO gene. So they turn this off. So when the CO gene is off, it's not producing this CO protein. That CO protein is not then able to turn on the FT gene, and we're not getting this FT protein. That's going to result in flowers. Right? Now, the expression of this CDF gene, so CDF genes, and there's multiple CDFs, CDF genes expression are regulated by additional transcription factors such as CCA1 and LHY. And this, again, are occurring in the morning, right? So in the morning, these genes are active. So the CCA1 gene and LHY gene are active, and this is regulated by these light receptors. And they produce these CCA1 and LHY proteins, which are transcription factors that then turn on the CDF genes. The CDF genes produce CDF proteins, which are transcription factors that prevent the transcription of the CO gene. And when this is not turned on, it doesn't produce CO protein, which doesn't turn on the FT gene and doesn't create flowers, right? So this is keeping the plant from flowering. Now, in the afternoon, when the plant can sense the changing daylight, we have the production of different transcription factors in the PRR family. And I'll just put I'll write it out. Transcription factors. Now these guys stop the production of CDF and CCA1 and LHY. Okay, so in the morning, these things are preventing the transcription of this CO gene. It's preventing the production of this CO protein transcription factor. But in the afternoon, the production of different transcription factors stop the production of the CDF genes, as well as the CCA1 and LHY. So it shuts this stuff all off, and as a result, we can start to have a buildup of our CO protein in the afternoon. So that's a good thing. That's going to start leading potentially to flowering. Now, this is all dealt with the really turning on and off of the gene, right? So these things prevented this gene from turning on to making protein. These PRRs turn these off, etc. We also have what is referred to as post-transcriptional regulation. And I'll get over to here. And we can do, whoops. So in the morning we had the production of all those CDFs, and those CDFs are preventing the CO gene, and these guys are preventing CO gene expression and CO protein as a result. And this is occurring in the morning, right? And then we said in the afternoon, we start to see a stop, and we reduce the production of these CDFs. But these proteins are still present in the afternoon. And they can still bind to those regions of DNA and prevent production of the CO protein. So we have some other sort of regulatory processes that are going on. And what is happening is the degradation of CDF protein is controlled by two other proteins, FKF1 and GI. And again, the production of FKF1 and GI is regulated by circadian clock. Okay, so we have these CDF proteins. They're preventing the expression of the CO gene. They're preventing the CO protein from being created, and that's preventing the flowering locus gene product from being created and preventing flowering. So how this works is FKF1 and GI are produced separately. But on long days, FKF1 absorbs blue light, and that activates it, and it allows it to then combine with GI. And it's this combination, it's this FKF1 and GI combination that will break down the CDF protein. So you need the combination to occur. How this factors in, how long days factor into this, is that this FKF1 and GI expression, or production, as I've been talking about it, overlaps on long days. Right, so on long days, once you get past that critical day length, that threshold, is when you're going to see this overlap. And when this overlap occurs, these things will combine and they will break down that CDF protein. And when we break down that CDF protein, it allows for the production of this CO gene and the CO protein and the production of the flowering locus protein. Now there's also some other things in play as well. So besides these FK1 and this GI, the CO protein itself whoops, I don't know how that happened. Itself can be stabilized or degraded by mechanisms related to blue, red, and far red light. And I'm not going to go into them because it continues to get more and more complicated, but essentially the CO protein itself can either be stabilized or broken down by various mechanisms that are related to day length and the type of light that you will see at different day lengths. So, to put it all together, we go back. In order for our plants to flower, our plants that respond to photoperiodism, particularly our long day plants, they need to express or produce this flowering FT protein from these FT genes. Now what's going to turn these FT genes on is a protein called CO protein. However, there are a number of things that prevent this CO protein from being produced. In the mornings, there is the production of these CDF proteins, which are produced by 2 additional transcription factors, and they prevent the production of the CO protein. Now, in the afternoon, there's a different family of transcription factors that stop these CDF proteins and allow for this CO gene to turn on and start producing CO proteins. At the same time, if we get to a long day situation for our long day plants, we have the production of both FKF1 and GI, and on those long days, their production overlaps. And when their production overlaps, these two will combine and will form this complex that further breaks down any CDF protein that had been produced in the morning. And as that protein gets broken down further and there's no more CDF protein that's being produced, then we see an increase in this CO protein itself, and that CO protein is going to turn these genes on, and we're going to see an increased expression, and then that is going to ultimately lead to flowering. All right, and now here is just a list of long day, short day plants and some day neutral plants. I don't think I had the day neutral included here. So these are just your long day plants on the left and short day plants on the right. Some examples and just some of their critical day lengths, just for your reference. All right, so we have self-inducive flowers, we have flowers that respond to photoperiodism, and then we have flowers that respond to vernalization. And I talked about vernalization before, remember? Right? These are plants that require exposure to low temperatures for flower induction. A lot of times these are those biennial plants that have that initial growing season, then they overwinter, and then they have the second growing season in which they mature and reproduce. but they may not all be. So different examples of plants that require vernalization: olives, winter wheat, Brussels sprouts, carrots, broccoli, hyacinth, tulips, lilies, all require this exposure to low temperatures before they will flower. Now, as propagators, you can do something called forcing, where you are going to artificially expose your plants to cold temperatures to force this flower induction. Typically, growers who may sell tulips or hyacinths in the spring will vernalize their bulbs in coolers to then trigger this trigger flowering at the proper time when they want to sell them. Okay, so that's flower induction, all of there. Now, flower development. So what's happening is your apical meristems, like I said in the beginning, are transitioning from vegetative to reproductive. Now, once this occurs, once that apex bud transitions, the process can't be reversed, right? So once it transitions to being a reproductive bud, you can't go back. However, the development of your flower can be aborted if conditions change, right? So if you had short day plants and somehow it returned to long day conditions, or if you had maybe extreme changes in temperature, your plant can abort those flower buds and not fully develop them. Now, if this occurs, you will have secondary or auxiliary buds that will continue with their vegetative growth until those conditions become right again for flowering. Easy. Flower development. Let's take a look at pollination. Now, again, we've looked at pollination already, so this isn't anything new for you guys. Pollination is simply the transfer of pollen from the anther to the stigma. And remember that anther is a part of the stamen, the male reproductive part of the plant that consists of the filament, the stalk, and the anther, and the pollen is created in the anther. And the stigma here is part of the carpel or the pistil, and it is the spot that receives the pollen. And then there's a style that leads to the ovary, and within the ovary are ovules that are going to turn into seeds eventually. Within each ovule is an egg cell and some other cells. So pollination is simply the transfer of that pollen to the stigma, and it occurs. There are two main types. So there are insect-pollinated plants, and there are wind-pollinated plants. With your insect-pollinated plants, typically they will have bright flowers or white flowers. They're trying to attract insects to them. Most of your fruit crops, vegetables, and legumes are insect-pollinated, and within them, in terms of pollinators, bees tend to be the most important as pollinators. But there are a number of other pollinators that are important as well, and we'll look at a couple of different ones. We won't go too in depth, but generally bees are the most important. Now, your wind-pollinated flowers, they're typically going to be inconspicuous, right? We don't need big, flashy flowers to attract anything, so they can be smaller, more inconspicuous, as you would see with some of our grasses and cereal grain crops, right? Those are wind-pollinated. They don't need big flowers to attract anything. So when I talk about pollinators, yes, bees are most important. Butterflies are very important as well, but then we also sometimes have some interesting ones. So hummingbirds act as pollinators. Bats are really important. Bats pollinate over 300 species of fruit, including mangoes, guava, and agave, which is important for tequila. Even snails get in on the action here. So this is the graceful awl snail, and that is a pollinator of a type of low creeping plant in the morning glory family. So it's kind of an interesting one. And there's an interesting relationship between these snails and bees that also pollinate these plants. But I'm not going to get into it right now. Essentially, when conditions aren't great for bees to be pollinating is when conditions are good for snails to be pollinating, and vice versa. So it's really beneficial for the plant because they're getting pollinated all the time. Okay, so pollination is transfer of pollen from anther to the stigma. Fertilization is the process by which the sperm cells within the pollen grain are able to fertilize the egg cell within the ovule, And again, we've looked at this in the past, right? Your pollen grain grows a pollen tube. A sperm cell will split into two sperm cells. That pollen tube grows into an ovule. It allows one sperm cell to fertilize the egg. The other sperm cell fertilizes what is referred to as these polar nuclei, and from that is the development of endosperm, right? And that's the storage, the seed storage. Some other things about pollination and fertilization. Some plants are self-fertile, so they can produce fruit and seed from their own pollen. They can pollinate themselves. Other plants are not self-fertile. They are either self-sterile, we'll refer to them as self-sterile, or self-incompatible. So they cannot pollinate themselves. They cannot create fruit or seed from their own pollen. And what can result from this process, if you have a self-incompatible plant that happens to pollinate itself, is something called parthenocarpy. So parthenocarpy is when a plant self-pollinates that is self-incompatible, right? So what is happening is these self-incompatible plants, they're not able to self-pollinate. There is a protein within the pollen that the plants can recognize as belonging to itself. And as a result, if that pollen does land on the plant, it will abort the developing seeds. So the fruit will develop as seedless. And we can see this sometimes in some of our navel oranges or some clementines. Could be nice if we are trying to produce these seedless varieties. Now, why does parthenocarpy exist in nature? One reason is we think it helps defend against predators, right? So in wild parsnip, up to 20% of the fruits are parthenocarpic. So these are seedless fruits, and they kind of act as a decoy for against seed predators, right? So if you are preying upon these parsnip fruits and you're preying upon the seeds, about 20% are going to be seedless, so you're wasting your time with them, and hopefully a couple fruits and seeds get by and get past these predators. They may act to, another theory is they may act to maintain seed dispersers. So if you're a plant and you are reliant on some sort of organism to help disperse your seeds, if you have a year where your pollination is unsuccessful, you may not attract those seed dispersers, may move on. But if you still produce a food crop, or you still produce fruit, those seed dispersers will eat the fruit and they will remain in the area for another season when you can hopefully have successful pollination. And like I said, seedlessness is a desirable trait for many human crops, and we've been able to cultivate parthenocarpic varieties as a result. All right, so we are moving right along. We are all the way up to fruit setting. So again, as a refresher, your fruit is your mature ovary plus associated structures, right? And in many plants, only a small percentage of flowers actually develop into fruits. Right, so your plants will produce a lot of flowers. Not all of them are going to develop into fruits. Many of those flowers don't get fertilized. Many may get fertilized, but they get aborted at the zygote stage. And one of the reasons this happens is because your plants may not have all the resources in terms of energy or nutrients to develop all possible fruits, right? It takes a lot of energy. There's a lot of resources that have to go into fruit development. And if you have too many fruits that you're trying to develop with your plant, you may overrun your resources, and either the plant may die or your fruits are going to suffer. Right? So if we are looking at fruit production, one problem is to obtain the optimal level of fruit set. Right? So you can have, we'll say, an apple tree, and you can have an instance where you have too heavy of a fruit set. So too many of your flowers have developed into fruits, And as a result, because of this resource allocation, your fruits are going to be undesirably small or of poor quality, or they may exhaust the tree's food supply. Right? So if you're too heavy, that's not a good condition. If it's too low, it may be unprofitable. Right? If you're only just growing, you have just one apple per tree in your orchard, that's not going to be profitable. So you want to try and find this balance. So as a result, usually half or more of the fruits are removed at a very early stage, and they can do this either by hand thinning or machine shaking, or using some sort of chemical spray. So I talked about apples. Like I said, they are typically thinned. Your apples tend to flower in clusters, right? And each flowers may develop into a fruit. So if you have a cluster like that, you will thin to about one fruit a cluster, and that fruit is referred to as the king's fruit. Just something interesting for you guys to know. All right, now this is a little primer for our next lecture, where we're going to talk about hormones. So with our fruit growth and development, there are a number of hormones that are important. They include auxins and gibberellins and some others that we'll talk about next lecture. So auxins are produced, for an example, how they are important. If you look at the strawberry, auxins are produced by the achenes, which are technically the fruit. And those achenes result in the growth of the receptacle, which is what we kind of consider as the strawberry. Right? It's the delicious part that we eat. Now, without those achenes, without the production of auxin, you won't have receptacle growth. So, but if you remove the fruits, they're saying seeds here, if you remove those auxins, and remove that, and you still apply a synthetic auxin, you will get receptacle growth. So it's just a little experiment I did showing the importance of auxin for growth, especially for strawberry growth. Gibberellins are another type of hormone that we're going to look at. Also important for producing bigger fruits. Particularly, they are used all the time with grapes. With your seedless grapes that you will buy at the store, almost all of them, probably all of them, have been treated with gibberellins, which is a plant hormone to make them bigger. Right? And you could see the difference between your treated grapes and your untreated grapes. So just about every grape you will buy at a store has been treated with gibberellins to grow larger grapes. Another important hormone is ethylene. Ethylene is actually the only hormone that is produced as a gas in your plants. So there are a number of fruits that are called climacteric fruits, and they release very high levels of ethylene as they ripen. And these include apples, bananas, tomatoes, apricots. And that ethylene gas results in, it's like almost like a positive feedback. It results or helps to ripen more fruit, right? So we can use a synthetic form of ethylene gas in a number of ways. We will use it to ripen fruits to a certain stage for sale, right? We may pick them before they're ripe and then treat them with ethylene gas, such as with tomatoes or bananas, potentially. And then we'll look at some other examples of how we use ethylene gas as well. But that's one of the big ones, is inducing ripeness. All right, so that's fruit set. And now we are going to end with aging and senescence of our plants, right? Senescence is just the deterioration of cells and tissues of an organism. Unfortunately, it occurs with every organism, except there is that immortal jellyfish that lives forever. It's actually really neat. It gets to a certain point in its life cycle, and then it reverts back to an immature, and then develops again and reverts back. it could theoretically live forever. So unless you're that jellyfish, you are most likely going to die at some point in your life. You are going to age and die. And that's what we're talking about. Right? So that aging process, that deterioration of cells and tissues is referred to as senescence. Now in our plants, they have different lifespans, just like other organisms, and they can be from a few months to thousands of years. Right? But eventually we're going to see senescence. And that may occur. When we're talking about senescence, we can talk about that in terms of the whole plant or the whole individual, or of particular organs such as leaves, flowers, or fruits. Now, we talked about vegetative growth patterns before, and we have our annuals where the whole plant is going to die after that first growing season. Herbaceous perennials where only the tops of the plants die, so we have that senescence of the stem and leaves as the growing season comes to an end. Same with your deciduous woody perennials where you have your leaves senesce and die off in the fall. Now, an interesting thing is we can extend the life of a plant through vegetative propagation. So, as an example, the black Corinth grape is a clone that has been grown in Greece for thousands of years. So as that plant is growing, your grape is growing, we will take a cutting from it that is still from an immature stem. We can then plant that cutting and it will grow a new grape, and it's going to keep technically the same genetics, the same kind of individual alive from centuries to centuries to centuries. There's some other things that you can do also when you take cuttings to grow them and kind of revert them back to a juvenile stage if you need to. It gets a little complicated. We cover it in plant propagation. But, so theoretically, you can keep a plant going through clones, potentially indefinitely, or until the sun explodes and destroys the earth. So, causes of senescence. It's usually the result of some inherent physiological change in the plant, right? They have evolved to have some lifespan, and eventually they reach the end of that lifespan, and there is some physiological things that are happening in the plant that are leading to that. But you can also have senescence occur as the result of pathogens or some environmental stress. But what's really happening, and particularly in leaf senescence, is all these things that are important for a leaf to survive are going away, right? So DNA, RNA, proteins, chlorophyll, photosynthesis, starch, oxygen, gibberellins, they all begin to decline, and that's leading to this death of this leaf. Often, particularly in your annuals, senescence is going to occur right after really after flowering and after fruiting, because your plant puts all this energy and all of these nutrients into the reproductive parts of the plant. Right? It puts it all into the flower, fruits, and seeds, and it does this at the expense of the root and shoot system. Right? Because it's the importance of reproduction. So as a result, you can postpone senescence in plants by picking off flowers early on before they start setting seed. Right? Because once they get to that process, they're going to put all their energy into reproducing, into developing seeds, and it's going to quickly lead to their death. Whereas if you just pick those flowers off before that happens, you can kind of postpone the life of your plant. And that's it. All right.